Hello and welcome to my presentation about our project with the title Personalizing Cancer Care, Integrating Data Modalities to Predict Overall Survival and Improve Outcomes for Patients with Non-Small Cell Lung Cancer. Our healthcare systems are under pressure to deliver better outcomes while reducing costs and increasing efficiencies. One reason for this is our growing world population, where people are living longer than ever before, and hence, chronic age-related conditions like cancer increase in their prevalence. Lung cancer remains the leading cause of cancer death worldwide, with an estimated of 1.8 million deaths in 2020 alone. And non-small cell lung cancer accounts for about 85% of these cases. We believe that we are at a pivotal moment in healthcare, where data, technology, and analytics provide opportunities to address healthcare challenges, accelerate research and development, and improve patient outcomes. Multimodal data increases our resolution of an individual patient's health status and her disease. Technology offers new ways of collecting data and enhancing care, and analytics provides us with new insights from data and technology. More accurate predictions of patient survival by integrating data modalities benefit both clinical research and clinical practice. In research, identifying high-responsive patients paves the way for enriched trial populations and the identification of predictive and prognostic biomarkers. Similarly, in clinical practice, where oncologists routinely make survival predictions to inform clinical decisions, more personalized models could enable the development of tailored treatment plans. To study the benefit from multimodal data integration, we investigated this in the Empower 150 trial. Empower 150 was a phase 3 clinical trial evaluating the combination of atezolizumab plus chemo and atezolizumab plus bevacizumab plus chemo against bevacizumab plus chemo as control. Interestingly, the atezolizumab arm did not demonstrate statistically significant longer overall survival in comparison to the bevacizumab arm, but the combination of atezolizumab plus bevacizumab plus chemo did, with a p-value of 0.02 and resulting in a 4.5-month increase in overall survival. Since adding atezo was improving the outcome, we wanted to know, can we identify a subgroup of patients that specifically benefit from the treatment with atezo and show significantly longer survival against the control arm? For this trial, we had H&E stained digital pathology slides and RNA sequencing data available that we wanted to investigate for their predictive utility. Besides the many successful use cases over the past years, where deep learning models were used to build predictive models on images directly, there is also a lot of potential to use them for annotation and subsequent feature generation. In this use case, we used the coordinates of tumor cells and lymphocytes identified by a deep learning model to generate the input for the development of spatial statistics algorithms. We investigated new ways to derive such spatial statistics features from the pre-called locations. These 41 features describe the tumor microenvironment of a patient on various cell levels, like intensity or distance metrics, as well as on the regional level, for example, in the form of prevalence maps. Using the spatial features alone already let us successfully identify the sought long survival group in the atezo arm, as you can see in the plot on the right. This comparison of predicted long survival groups had a hazard ratio of 0.64 and a p-value of 0.012. So the question remained, can we further enhance the prediction by adding a second modality, namely gene expression data? Before we look at the results, let's look at the graphic of our feature selection and data integration pipeline first. This is a pretty busy slide, so please bear with me and it will become clear. From a bird's perspective, this is a Monte Carlo cross-validation pipeline, used in line with an FDA guidance for predictive enrichment analysis. That means we are performing 1,000 train, test, and validation splits, and the category of each patient, be it long or short survivor, is decided by a majority vote across all of these 1,000 models. The survival plot 2 slides before showed a comparison between these predicted long survivors of each trial arm. In the middle of the workflow, all spatial features are mixed with selected gene expression features. A rich Cox regression model is built on the atezolizumab training set, tuned for an optimal difference to the bevacizumab arm on the validation set, and then a prediction on the test set is performed and stored for later. We have tried many different approaches to include gene expression data while keeping individual gene resolution for subsequent biomarker identification. This turned out to be a difficult task, maybe due to inter-patient heterogeneity. Ultimately, we constructed a stability selection approach where we bootstrapped from the training set 1,000 times in every of the 1,000 iterations and use a 10-fold cross-validation Lasso-Cox model to select a small subset of genes. Each iteration, we store the list of the identified genes and use the top n genes then to be passed on and integrated with the spatial statistics features. The number of passed down genes was another hybrid parameter of our model, but the best performance was at around 7 or 8 genes. To acquire the empirical probability of a gene being selected more than n times, we ran the whole pipeline 1,000 times with permuted gene expression data. You can see for the individual modality performance, patients that had both digital pathology and gene expression data available show a significant median survival difference in favor of the predicted long surviving atezo recipients. On this slightly smaller set of patients, the spatial features-based model had a hazard ratio of 0.61 and a p-value of 0.013, resulting in a 7.1-month increase in median overall survival. Gene expression features alone also led to a slightly significant model outcome with a hazard ratio of 0.64 and a p-value of 0.042, resulting in a 5.3-month increase in median overall survival. Combining the two modalities resulted in an even more pronounced difference in terms of a reduced hazard ratio of 0.44 and a smaller p-value of 0.001, resulting in a 6.7-month increase in median survival. The biggest separation between the survival curves in comparison to the spatial features alone is visible in the first 3 to 6 months. These data show the complementary value of both data modalities in the prediction of overall survival. Here you can see the most frequently selected genes by our stability selection pipeline. Interestingly, only 2 genes were selected at around 50% of all iterations. This might be indicative to the already mentioned patient heterogeneity. Please keep in mind that the frequency of the gene being selected does only say that it stably contributed a signal in many patient subsets, but not that it was the strongest signal of each model. The plot on the right shows the negative correlation that MAML3 has for atezo, where no such correlation can be observed for bevacizumab. In terms of biological interpretation, these genes seem to be involved in various biological functions. For example, MAML3 is a member of the Notch pathway, and AC024475 is a link RNA. Besides the predictive utility, these findings need to be validated on a different data set, and if consistent, further research is required to understand their function. With further validation, our model could impact clinical research and clinical practice. In terms of research, it could enable smaller, more efficient clinical trials with enriched patient populations, with potential to generate deeper insights into individual treatment responses and the identification of predictive biomarkers. In clinical practice, it could inform the development of companion diagnostics to predict individual treatment response and also supporting clinicians in making more informed, personalized care decisions in the future. And of course, patients, by improving the standard of care and helping more patients to get access to the right treatment at the right time. This is our vision of personalized healthcare at Roche. We are applying rigorous analytics to high-quality data sets and utilizing novel technologies to create a continuous cycle of insight generation from bench to bedside and vice versa. By combining insights from the real world with novel analytics and technologies, we are generating deeper insights into disease and transforming clinical trial design, for example, with virtual control arms, for more sensitive clinical study endpoints, to predict innovative new treatments to patients faster than ever before. We are leveraging insights from R&D to develop evidence-based, value-driven healthcare solutions that enhance our ability to deliver personalized diagnosis and care, enable faster access to treatment, and support digital disease monitoring. Ultimately, by unlocking the potential of data, technology, and analytics, we can create more resilient, sustainable healthcare systems that deliver benefit and value for all of society. So if you're interested in that, please visit our booth at AMLD to find more about our personalized healthcare approach. Finally, I would like to thank the patients and their families who took part in the Empower 150 trial. I would also like to thank the investigators and clinical study teams and our team at Roche Genentech, Gunther Janssen for the initial ideas, scientific input, and project support, Teresa Carra for developing the gene expression pipeline, Xiao Li for the development of the spatial statistics approach and building the algorithmic foundation, and the imaging team for their pre-work on image quality and annotation. Thank you very much for your attention.